So I'm going to talk about optimizing linear estimators of single trial fMRI responses. And this is work done together with Scott Lee and Hunar Abdul Rahman. So sometimes we want to estimate responses to individual trials in event-related fMRI. For example, if we want to use something like beta series regression, which is simply the correlation between trial-by-trial estimates across or between two ROIs. Or more commonly, we might want to look at patterns across voxels for individual stimuli presented on individual trials, as in MVPA or RSA. But as you know, the time between trials, the stimulus onset asynchrony or SOA, is often shorter than the fMRI bold response, which can last up to 30 seconds. So we need a method for linear deconvolution. And we do this in the context of the general linear model, or the GLM. And a standard approach that Mumford et al. Called least squares all, or LSA, is simply to model each individual trial as a separate regressor or column in this design matrix, where the rows are data points for each TR or each scan, and the light and dark colors here indicate an HRF in response to individual trials. And from that model, we get an estimate, a beta, for each trial. In 2010, Turn introduced an alternative called least squares separate, or LSS. In this case, a separate GLM is fit for each trial. So when you're trying to estimate trial one, you have one regressor for trial one and a second regressor for all other trials. Then you re-estimate a new GLM for trial two, etc., where all the trials that you don't care about are modeled in a second regressor. Takes a bit longer to run, but it's very easy to do on ROIs. And if you want some code for it, there's some code in GitHub here to compare LSS with LSA. In 2016, Hunar and I wrote a simulation paper, where we simulated two sources of variance: variability from trial to trial in the betas, and variability from scan to scan, or the noise. And we then looked at the efficiency of estimating the betas using LSA and LSS as a function of the time between trials, or the SOA, and the ratio of trial to trial to scan variance. And we also looked at the spatial correlation of that variance across voxels. But what we didn't look at is the effect of temporal autocorrelation in both trial by trial variance and scan by scan variance. And that's what I want to show you today. We also didn't consider non-random ordering of trial types. So in that paper, we assumed you had a randomized design. And I'll show you some data when the conditions have to alternate, for example. So another example of temporal autocorrelation in your actual design. And finally, that was a pure simulation paper. So what happens with real data? There we don't know the true betas, but we can still estimate the efficiency of estimating various things. And I'll show you some real data at the end. But the simulations that I'm going to show you are based on a real paradigm from Wakeman, in which there are 19 participants and 9 runs with an SO of 3 seconds, a TR of 2 seconds. There are actually 9 different conditions, but I'm going to focus on 2 contrasts. Firstly, we can compare faces versus scrambled faces. And for these, the ordering of faces and scrambled faces was effectively random. So that's a random contrast, but we also had immediate repetitions. So if you want to compare the first versus second presentation of the same stimulus, face or scrambled, then that's an alternating contrast in your design matrix. And if you want to get the data, they're here. So how do we measure efficiency? Well, we have these 2 estimators, and they give us a beta for every voxel. That's the columns here, for every trial in a particular condition C. And then we get that on multiple runs. If we wanted to calculate the precision of estimating the single univariate response, so the ROIs average over voxels and over trials, the basic idea is we simply take our estimates, we average over voxels and trials, and look at the standard deviation of that across runs. So a better estimator is one that has a lower standard deviation across runs, or a higher precision. I'm not going to talk about this univariate estimate today, but the same idea applies to looking at the precision of beta series regression. So now you have 2 ROIs. We're going to average over voxels. So now we just have a single beta per trial, and we get a correlation for each between the time series, or the beta series, for each run, and then we just look at the standard deviation of across runs of these correlations. And finally, if we're talking about MVPA, here there are several measures. I'm going to talk about one of them, between run classification performance. Basically, here we're going to average over trials, but have a pattern over voxels. And then we have the same condition in multiple runs, so we can look at how similar the patterns are for the condition 1 across runs, and compare that to how similar condition 1 is with condition different condition across runs. And generally, classification performance would be better when the within condition comparison is higher than the between condition correlation. So let's look at some of these metrics, starting with the precision of the sample correlation as relevant for beta series regression. And these are simulation results where there's no spatial or temporal correlation in the variability. So plotted here is efficiency, where lighter is better. We've got LSA and LSA, sorry, LSS and LSA on the left. On the top, we have a situation where the trial-to-trial variance is higher than the scan-to-scan variance. On the bottom, we have when the scan-to-scan variance is higher. As you can see, which is a better estimator depends on the ratio of those variances. When the trial-to-trial variance is higher than the scan-to-scan variance, LSA is better, and the opposite is true when the scan noise is higher than the trial-to-trial variance. Why is that? Well, we looked at this in the original paper. The reason is that LSS is effectively applies a temporal smoothing. It's still an unbiased estimator, but it achieves its efficiency by smoothing over time. And that's why temporal autocorrelation is very relevant, which I'll come on to later. But to explain this first finding, here are the true betas are in green, and the estimates from LSS are in red. And you can see that they're slowly varying, the LSS estimates, and they don't capture the high frequency variation. This is when the trial-to-trial variability is quite high, and LSA can more accurately capture this high trial-to-trial variance in the betas. When the scan noise is higher, though, LSS does better by effectively smoothing over high frequency variations in the noise, whereas LSA basically confounds the scan variance with the trial variance. So it confuses noise in the data with true trial variation. And you can see the blue lines now are less efficient. And that's why the relative efficiency of these two estimators depends on the ratio of trial variance to scan variance. What about when we're doing MVPA and we're interested in the and when might be spatial correlation between the voxels? Sorry, we're still talking about beta series regression, but now there's spatial correlation in the data. For beta series regression, it doesn't matter because you're going to average over voxels anyway. But for MVPA, if we took out between run classification performance measure, you can see which is the better estimator again flips. So now when there's spatial correlation in the trial-to-trial variability, LSS becomes better. Or when there's spatial variation in the noise, LSA becomes better. Well, why is this? Well, again, we simulated this in our original paper. If you're interested in pattern classification, what's important is the difference between voxels, because the pattern is essentially a difference across voxels. So the red and blue here are two different voxels. The first 25 trials are condition 1, and the second 25 trials, or sorry, 30 trials, are condition 2. So if we take the difference between voxels, that's the black line here, classification performance is basically measured by how big the difference is between conditions in this mean here relative to the variability of the conditions of the pattern difference or the voxel difference. So here LSS is quite good at classification, whereas LSA, if you now look at the difference in means relative to the spread within a condition, you can see classification performance is worse for LSA when there's spatial correlation of the trial variability. But that again flips when we have spatial correlation in the noise. And the reason for that is if you take the LSA estimates, I showed you previously, these vary very rapidly when there's noise in the data, but because that variation is the same for all voxels, once you subtract voxels, that variability disappears, it's cancelled out, and you get high classification performance. So when the spatial noise is highly coherent across voxels, LSA can still estimate patterns very well. But let's come to new stuff now. What happens if there's temporal correlation in the variability across trials or in the variability across scans? Now, for this is the beta series regression, the precision of sample correlation. And now, again, the better estimator has flipped again. So now it's LSS is better when there's temporal correlation in the trial-to-trial variability, whereas LSA is better when there's temporal correlation in the noise. It's a bit harder to explain, but let me try to explain. So again, the green are the true betas, and the red are the LSS estimates here, and the blue are the LSA estimates. The R here is the correlation coefficient between the true betas and the estimated betas, so higher is better. And you can see that when the trial temporal covariance or autocorrelation is higher than the scan autocorrelation, LSS is better. In the black are the actual trial-to-trial data, which I've just shifted down a bit to make it easier to see. Compare that case where we add temporal correlation to the noise. You can see the data now, the scan variability is smoother. What happens, if you just about see this, is that the LSS, because it temporally smooths things, when there's temporally smooth noise anyway, it again starts to conflate that with true trial variability. So you can see some low frequency drift in the LSS estimates away from the true values, which is why its correlation is less than with LSA. So again, because of the temporal smoothing properties of the LSS estimator, if the true betas are temporally smooth, or the noise is temporally smooth, that affects its relative efficiency. And that's the reason we did these simulations in the first place. Finally, if we look at the effect of temporal correlation on between one classification performance, or more MVPA, here we have a first situation where the efficiency depends now on the particular contrast. So whether it's a randomly ordering set of trials, as in face minus scrambled contrast, or an alternating contrast of first versus second presentations, that affects which is a better estimator in the case of temporally correlated noise for LSS versus LSA. And again, we can look at the difference between voxels to try and understand how the temporal correlation affects these two properties. But if I just flip forward and backwards between what I've shown you, the first thing you can see is as I flip backwards and forwards, the relative efficiency of these two estimators changes. So the first thing it depends on is the question you're asking, whether you're interested in beta series regression or MVPA. Second thing it affects it is whether the trial variance is higher than the scan variance, and also whether that trial variance is spatially or temporally correlated. And finally, the particular contrast, the ordering of your conditions, also has an impact. So if you're interested in beta series regression and there's independent noise, then LSA is generally better than LSS when the trial variability is higher than the scan variability, and vice versa. For spatial covariance, sorry, my tabbing has failed here. The LSA is better, but that switches when the trial variance is higher. And this is independent of the trial order. For MVPA, LSA is better, like for beta series regression, when the trial variance is higher. But when the spatial covariation across voxels, then the efficiency swaps again. And finally, when there's when you have temporal correlation in the beta series and/or the noise, then the efficiency depends on the contrast. So all these factors affect which is a better estimator for your single trial responses. But what about real data? So just to finish up, the first thing we did is went to the Wakeman data and we asked the question, is there significant variability from trial to trial in these data? So for each voxel, we fit a design matrix where the first 9 columns are what we call LSU, which I haven't talked about today, but that's the standard approach where you model each condition separately, but all the trials within that condition are concatenated into the same regressor. So it just captures the mean difference between conditions. And then we added another set of columns which modeled individual trials like LSA. And then with an F contrast, we can ask where in the brain is a significant variability from trial to trial over and above the difference between means of the conditions. And here are the 19 subjects. In the bottom right is a group analysis. And you can see this is family-wise error corrected. There are many voxels across the brain that show evidence of trial to trial variability that's above the difference in the conditions between the difference in the means between conditions. The bottom right is a meta-analysis of statistics, and you can see this is again corrective model comparisons that, particularly in parietal cortex, there's a lot of variability from trial to trial. And so these are the simulation results that I've shown you already. Let's add the results from 2 ROIs. So we took the fusiform face area because this is a paradigm with faces. So this is, we know this responds generally to faces. And we took the posterior parietal cortex, which is this region here, which showed evidence of high trial-to-trial variability. And both these cases, for beta series regression, LSS was more precise or had a variability in the estimate across runs than did LSA. We don't know the true betas for data, but we can talk about the efficiency in terms of the variability across runs. If we look at MVPA classification, here there wasn't actually much of a difference between the two estimators, although for FFA, LSS was better when you have a randomized contrast. A second example, very quickly, this is from CanCan, thanks to Ethan Knights. We were decoding the finger, so people pressed one of 4 fingers on their right hand, and so this is the contralateral hemisphere of motor cortex, and you can see that with LSA, classification was at chance, 25%. It was only with LSS that we managed to get classification performance above chance. And as you expect, the ipsilateral motor cortex didn't show such classification. So here's another situation where LSA does appear to be doing better than LSA. Sorry, LSS seems to be doing better than LSA. So LSS so far seems to be better, although there's one paper published showing that LSA can perform better in other cases. But what it's suggesting is that in the data we've looked at, either the scan variance is much higher than the trial variance, and or the scan spatial covariance is higher than the trial spatial covariance, and or the trial temporal covariance is higher than the scan temporal covariance. We can't tell which of those is the case, but these are factors you might want to consider. There's some caveat which I'm about to address in simulations is at the moment the data have been pre-whitened, and that might affect the results. But this may be something to consider in your analysis when you're looking at single trial responses, such as for MVPA or RSA. Thank you.